Hello. The topic of our lecture is biology of cancer. In this lecture, we're going to talk about how cancer cells differ from normal cells, and I do expect that you'll be able to pick out those key differences. We'll talk about the genetic basis of cancer, specifically looking at oncogenes and tumor-suppressing genes, as well as the role of inflammation in the development of cancer. It's important that you be able to define some of the origins of cancer based on very common nomenclature or very common definitions. And this will be your responsibility as identified there on the slide. We're going to look at tumors that produce biologic markers and then connect those biologic markers to some very specific tumors. There isn't any way I expect you to know or memorize every tumor marker, but there are some key markers that we'll talk about that you should have an understanding of. We know that the pathology of cancer can produce manifestations, symptoms, and so we'll look at some very general manifestations of cancers and then also take a look at the core concepts of metastasis as well as some mechanisms and patterns, and then also staging of cancers using the tumor nodes metastasis staging system. The very last thing we'll talk about are a few things to remember when we think about different age groups and the development of cancer. So let's start off by talking about characteristics of cancer. Well, we know that a cancer is a malignant tumor or neoplasm. Now, it's important to remember that not all tumors, even though they're not normal, are necessarily malignant. So that then leads us to be able to differentiate between a benign and a malignant tumor because they do look different. A benign tumor is filled with encapsulated and differentiated cells, and these type of cells do not spread. Importantly, when you look at a benign tumor, the cells within those tumors, because they are encapsulated and they are differentiated, tend to have the characteristics of normal cells, which means they basically play by normal cell rules. This is very different than what happens with malignant cells. Malignant cells have altered structures, they grow faster, they spread, and they do not have characteristics or look like normal cells. They don't play by normal cell rules. When we think about then the loss of normal cell characteristics, what we're talking about is aneuplosia, and this aneuplosia is key. It means that these cells are not differentiated. The nucleus tends to have an irregular shape and size, and this then results in tissues in whom structures become abnormal. If the malignant cell has a capsule, it has very little integrity. But these anaplastic characteristics, or the characteristics of malignant cells, the ability to have different structures, grow faster, and spread, allows the cancer to actually spread to other structures like vessels and lymphatics. So if we if we kind of compared and contrasted benign versus malignant tumors, you can see that benign tumors tend to grow slower than malignant. They have very well-defined capsules, whereas malignant tumors, as we mentioned previously, are not encapsulated. Benign tumors aren't invasive, where malignant tumors are. Very well differentiated cells on the benign side, but very poorly differentiated, or that concept of aneuplosia, on the malignant side. Now, one of the things I mentioned previously is that they tend to grow faster. So as a result, then, benign tumors have a very low mitotic or reproductive index, as opposed to malignant tumors having a very high mitotic index. And then finally, benign tumors do not metastasize, whereas malignant tumors do have the ability to spread beyond their initial area of growth. They can spread distantly, which is metastasis. Let's talk then about some of the behaviors of cancer cells. First off, cancer cells have become that way. These are normal cells that have transformed and become cancerous. These cancer cells behave autonomously, and as a result, they don't follow the usual cell control mechanisms. So what are some of those things that are very important that are different? First off, cancer cells don't have contact inhibition. Normal cells will decrease their growth when they start touching other cells. Cancer cells don't do that. Because they don't have this contact inhibition, when they come up against or contact other cells, they continue to grow. Normal cells would stop growing. Another mechanism is that they don't need an anchor for continued growth. As a result, then they're able to grow in a suspension. Normal cells don't have that ability. And last but not least, cancerous cells have a long lifespan. They don't have programmed cell death or programmed apoptosis. So they're going to live longer than the normal cells have the ability to do. As mentioned previously, cancer cells also lack differentiation, this characteristic being called anaplasia. Because they lack differentiation, they don't acquire specialized functions or organization, so they don't become a functional cell. These cells look very, very different. The size and the shape isn't uniform, so that means they're pleomorphic. That means different sizes, different shapes. Normal cells in tissue cells within a defined tissue type all tend to have the same size and shape. Cancer cells don't do that. They all look very different. The more non-differentiated the cells are, the more malignant they are. There is a great book that actually looks at the history of some of the things that we know about cancer cells. And this is a book called The Immortal Life of Henrietta Lacks. Henrietta Lacks was a woman of color in, I think it was the 30s or the 40s, and she had uterine cancer, and she was treated and followed up at Johns Hopkins Hospital and Johns Hopkins Medical Center. At that time, we were just learning a lot about cancer cells and how they actually behave, reproduce, and how normal cells become cancerous cells. Well, this cell line from Henrietta Lacks actually became the first cell line that we continue to reproduce over and over and over as a cancerous cell as we learned more and more about how cancer cells develop, grow, and behave. So we owe a huge debt to Henrietta Lacks. Some other cancer cell behaviors that are important to understand are the metabolism and how these cancer cells metabolize. Remember that these cancer cells are rapidly dividing. And as they are able to rapidly divide, one of the things that they're able to do is actually survive in a less than optimal environment that normal cells wouldn't tolerate. Cancer cells will put up with an acidotic, hypoxic environment. Normal cells don't do well in that type of environment, but cancer cells are more adaptive. They are rapidly dividing, and so they have the ability to survive in less than great environments. Because these cells are rapidly dividing, they also use nutrients that are then not available to normal cells. So essentially, our normal cells, because they are dividing more slowly, don't have the necessary nutrients because the cancer cells are taking up all of that nutrition that the normal cells would need. At the same time, these cancer cells have the ability to form glycolysis without using mitochondria to generate the usual ATP. At the same time, though, they still can generate a lot of ATP. Normal cells use the process of oxidative phosphorylation to turn glucose into CO2 and energy, or ATP. Cancer cells don't use that normal process of oxidative phosphorylation. And in doing so, they consume huge amounts of glucose that then continues to support their proliferation. Now, despite this abnormal pathway and even able to use an anaerobic metabolic pathway, they are able to form lipids and other cell building blocks because they're using a lot of that glucose. Because they use that anaerobic metabolic pathway, they are using lactate, and they may have some they frequently display an incomplete breakdown of that glucose, and that a lot of that also is because they don't use that oxidative phosphorylation pathway. So by being able to change the way they metabolize, they are able to outgrow their environment. They don't put the brakes on. And this switch to a different way of metabolizing is encouraged by or aided by the cancer cell genes themselves. So what are some other behaviors we should be aware of? Well, we should be aware of stem cells versus cancer cells. In normal stem cells, remember stem cells are immature cells. In normal cells, these stem cells are constantly replacing themselves, and they regenerate using adult stem cells. Normal stem cells are and pluripotent, which means they have the ability to replace themselves and differentiate to a wide variety of cells. Cancer cells also have stem cells or immature cells. They have some similar abilities as normal cells, but they produce a very limited number or types of cells. Therefore, the key concept here is that normal cells, normal stem cells, have the ability to differentiate into a wide variety of cells. Cancer stem cells do not have the ability to produce a wide variety of cells. They produce limited variety, a limited type of cell, and that limited type of cell is that abnormal cancer cell. It's important to know what we mean by carcinoma or cancer in situ. In general, when we look at carcinoma in situ, we're looking at a pre-invasive cancer, and these are atypical cells. These atypical cells proliferate faster than completely normal cells, but they have not penetrated the basement membrane, therefore they do not have local invasion. So as a result then, when we look at cancer cells in situ, these are not malignant cells, but they're not normal cells either. So what might those abnormal cells do? So they can usually do one of three things. They may stay where they are and not change. And while they're staying where they are and not changing, they're essentially waiting for the right time or the right environment to come together. They may actually progress to invade that basement membrane and metastasize. When they do that, they become cancer. But the other thing they may do is regress and go away. So the immune system may locate them and start breaking them down. Or because the environment just never seems right, or the time just never seems right to those cancer cells, they will gradually regress and go away. Now, a general rule of thumb when we think about carcinoma or cancer in situ is the more dysplastic the cells are, so the more abnormal the cells are, the more likely they are to become invasive. So what do we do with this information then? Well, in our common health care, we have two choices. We can either remove them or we can monitor them. And the level of dysplasia may determine whether or not we decide to do some removal or whether we continue to monitor. And the great thing about the monitoring is one of the ways we may be able to monitor them is through tumor marker surveillance. Because remember, cancer in situ is not malignant. they're not normal, but they're not malignant. So if you have dysplastic cells and there's a biologic tumor marker that you can use for surveillance, you would increase that tumor marker surveillance to see whether or not these cancer cells, these in situ cells, are becoming cancerous. Let's take a look then at biologic tumor markers. When we look at biologic and these tumor markers, we have to remember their purpose and their use. When we look at those markers, the purpose of following and measuring tumor markers is for surveillance, diagnosis, to guide our therapy, to assess recovery, and or to assess recurrence. So a good example of that would be CEA, our carcinogenic embryonic antigen. CEA, that tumor marker, will actually decrease with tumor, but you can with treatment rather. But if you continue to follow that marker as a not only a recovery, so let me start over. With CEA, with this which is the carcinoembryonic antigen, CEA, it's so much easier to say CEA. We use that for surveillance of certain types of cancer. When that marker, when the level of that marker goes up, we're starting to look at it in terms of additional diagnostics to take a look at. But once we begin our treatment, when that CEA biologic marker begins to decrease, it allows us to assess the response to therapy. So we know whether or not our treatment is working. After an individual has been treated, then we can continue to use the marker to look for increased activity, which may tell us whether or not recurrence of the cancer is there. Now again, these biologic markers may be produced by both benign and malignant tumor cells. So we just have to know our marker. Generally, these markers are found on or in the tumor cells, in the serum, or in other body fluids like the spinal fluid or the urine. So if you have a tumor marker that is found on or in the tumor cell, it could be a hormone, an enzyme, it could be a gene, one of the antigens, or even an antibody. When we look at fluids that we're looking at, we're actually looking at fluids that could actually transport those cancer cells. So this could be in the spinal, as we mentioned, the spinal fluid, or in the urine. And finding tumor markers in the urine, I think we talked about that in terms of multiple myeloma and looking for the Bence-Jones protein. Now, if the marker causes symptoms, it can actually, these symptoms are called the paraneoplastic syndrome, and we're going to talk about that a little bit more shortly. So what are some of the biologic markers that we use? There's a huge and growing number of biologic markers as we learn more and more about cancers and the cellular activities of cancers. But what I've done here is given you a key to some of the more common and those that we have a fair amount of experience with. And so let's just sort of look at some of the links of the markers to some of the tumors that we're aware of. So if we look at alpha-fetoprotein, this is a serum marker that we use for surveillance of liver and germ cell tumors. PSA has been around for a very long time, and this is prostate-specific antigen, and this is a marker for prostate tumors. Now, increasingly, PSA has come under fire as an absolute marker, so we look at an index and we trend that over time. CEA, as I mentioned earlier, is a very common marker for a large number of tumors, and so you'll see an elevated CEA in colon, breast, lung cancers, so a number of different markers. And CEA is great example of looking at sensitivity versus specificity. So when we look at CCA, it's a fairly sensitive marker, which means it will pick up a number of different types of tumors. So it's pretty sensitive in picking up cancer cell as a cancer cell marker. However, it is not specific because it doesn't tell us exactly which tissue is being invaded by the cancer. As I mentioned, and you can see it on the slide, so CEA will go up with colon, lung, breast. So even though it is sensitive, it's picking up the cancer, it doesn't tell you whether it's colon or lung or breast. Bench-Jones protein, as I mentioned, that's a urine marker for multiple myeloma. The BRCA1 and BRCA2 are gene mutations. And this is one of the first gene mutations that we identified in terms of cancer markers. And this is breast cancer 1, breast cancer 2. Most of our experience is with breast cancer because that's where we identified it. However, we know that BRCA1 and BRCA2 are also implicated in ovarian cancer. So we want to look for that as well. In addition, newer data tells us that in males who have a family history of women in their family of having BRCA1 and BRCA2 breast cancer may have an increased susceptibility to prostate cancer. Another cancer marker that we've looked for is neuron-specific enolase, or NSE. This is a serum marker for small cell lung cancer and neuroblastoma. So again, looking at those ideas of sensitivity versus specificity. Now the website that I've given you are listed on the slide is really for your information. For your future and for your interest, because this is a great fact sheet and one of those things that I think you'll find useful in your practice, either now or in the future. Other issues or concepts to consider in the biologic cancer include the genetic and inflammatory basis of cancer. So let's start by looking at some of the issues with the genetic basis of cancer. When we think about the genetic basis of cancer, we're looking at the changes in the cell's genes that actually cause the cell to become a cancer cell. Because remember, when you have a cancer, it starts as a normal cell that becomes a cancerous cell. So what happens in that cell's genes to cause it to become a cancer cell? So we're looking at changes in the DNA and in the RNA. So one of the basic theories in the genetic basis of cancer is that these gene changes occur fairly consistently and constantly over time, so that when a critical number of mutations occur within that cell, then that cell becomes cancerous. That's the idea then of stepwise accumulation. We see that in skin cancers. When you think about a skin cancer, the skin cells are exposed to ultraviolet light over time and frequently. And so just simply being exposed to the ultraviolet light doesn't make all of our skin cancers become cancerous, but something does trigger over time, and with a lot of accumulation, those skin cells may then develop altered genes, and those altered genes cause the skin cancer. As those mutations occur and cells become a cancer, then those cells have an advantage over the normal surrounding cells. And what were those advantages? Shout at the screen. Yeah. They don't have contact inhibition. They develop angiogenesis, and they have a rapid metabolic rate that consumes nutrients. Because they have advantages over normal surrounding cells, they are able then to proliferate, and that's that clonal proliferation. They're making lots and lots and lots of copies. And then initially maybe a cancer in situ with then the potential for advancing. So what happens then is normal genes can then become oncogenes. So let's take a closer look at oncogenes. Oncogenes then are genes that have mutated, and because they have mutated, they now direct the synthesis of proteins that accelerate their proliferation. The idea here is that some mutations drive the cell to become a cancer. So when these cells, these oncogenes, these genes that have mutated and now changing the synthesis of proteins, they may amplify, they make numerous copies of these mutated genes, and that becomes important both in the prognosis and therapy, because if we can turn off these mutated genes, if we can turn off these oncogenes, then we can stop the synthesis of these proteins. And if we stop the synthesis of these proteins that cause the acceleration and proliferation, you no longer have the mutations driving these cells to be cancerous. Now, normally these oncogenes are balanced by tumor suppressing genes. Tumor suppressing genes are genes that inhibit or decelerate the proliferation of these abnormal cells. So when tumor suppressing genes are active, they actually suppress the oncogenes. And because they're suppressing oncogenes, they decrease cell growth, they decrease mutation, they decrease abnormal replication. So if you take those two ideas together, if you don't have suppressor genes, you essentially activate oncogenes and therefore progression to cancer. You want those tumor suppressing genes to balance out the oncogenes. The end result of the gene changes, if you have negative gene changes, is you lose the caretaker gene functions. And one of those functions of our caretaker genes is programmed cell death. The second is that contact inhibition. Now, the other thing to remember is that these gene mutations generally occur in somatic cells, therefore cannot be inherited. However, predisposing mutations are inherited. So what we mean by that is predisposing genes are BRCA1, BRCA2. As opposed to someone who has a lung cancer, we know that in general, lung cancer, which is a somatic cell, is not a mutation that passes through families. So if we take all of that together and we look at gene misregulation, when we think about this misregulation of how gene changes foster the development of cancer, we know that these cancer genes, genes in cancer cells, have the ability and do secrete growth factors to stimulate their own growth or to increase growth receptors. We know that these cancer genes don't respond to growth stopping signals. I keep coming back to this idea of contact inhibition. We've talked about that apoptosis, programmed cell death, has been disabled. Because of the changes in genes, these cancer cells are able to produce new blood vessels, and that's the idea of angiogenesis. Normal cells don't have angiogenesis. Cancer cells do. We know that cancer cells have the ability to divide multiple times, as opposed to the limited number of times that normal cells are able to reproduce. So you think then about cancer cells having a level of immortality. And then last but not least, this gene misregulation supports metastasis. Cancer cells have the ability to move away from their original site where they were first developed. And they're able to do that because they have less adhesion. And remember, they have that ability to survive in new and different environments. So I hope we pulled some of those concepts together in looking at the difference between normal cell behaviors and cancer cell behaviors. So what about the role of inflammation and the immune basis of cancer? We know that the goal of inflammation in the immune system is to recognize that cancer cells are antigenic and then destroy them. So they have to be able, our immune system has to be able to recognize cancer cell surface antigens and therefore be able to destroy them. Inflammation is both protective, that ability to recognize and destroy, or it can also be potentially harmful. And this is what we see in HPV and the role of inflammation in the human papillomavirus in becoming cervical cancer, or the risk of hepatitis B and C becoming a liver cancer. We also know that the inflammatory and immune system plays a very important role in cancer progression. Cancer cells actually are very, very opportunistic. And as a result, because of their opportunistic nature, they actually use that inflammatory response to their benefit. And they do that because they use healing and proliferation pathways to support their own growth. Cancer cells have the ability to produce chemotactic factors. Those chemotactic factors or signals attract macrophages. Once they attract those macrophages, though, they actually produce mediators which then decrease inflammation, increase proliferation, and increase angiogenesis. Biologic adaptability of cancer cells is absolutely critical to the understanding of how cancer cells are able to keep going. They are ego-driven survivors. Remember, they will change their environment to support their growth. And their ability to attract macrophages and sort of reprogram those macrophages in order to decrease the cytotoxic response and not kill cancer cells is a big part of that. At the same time, cancer cells have the ability to change the T regulatory cells. When they change those T regulatory cells, there is no cell destruction, as well as they produce cytokines that allow them to continue to support tumor cell proliferation and tumor cell spread. Without the T regulatory activity, tumor cells are not destroyed as an end result, because again, they change those T regulatory cells to protect themselves to not be destroyed and allow them to continue to proliferate and allow them to continue to spread. So then how can we link what we know about the behaviors of cancer cells to how cancer manifests? And there are just some general things we'll talk about. First off is this idea of the paraneoplastic syndrome or syndromes. The concept here of the paraneoplastic syndrome or syndromes is that these are symptoms that are triggered by cancer, but they aren't due to the mass effect of the tumor itself. So if you get a big pancreatic cancer, you will have mass effect symptoms like pressure, pain, changes in the function of the organ itself. These are actually symptoms that are triggered by the cancer, but they're related to the biologic substance like hormones that may be released from the tumor or from the immune system. So why does this matter? Well, because these may be the first symptoms of the cancer that we notice. So the paraneoplastic syndromes can be very broad-based, and may help us actually give a sense that something is wrong and we need to do further investigation. There's a good table in your text, table 12.4, that takes a look at this concept of paraneoplastic syndromes. One of the most common manifestations of cancer is cachexia. And cachexia is energy intake being less than energy expenditure. So a lot of expenditure, not a lot of energy intake. The way this frequently is manifested is through weight loss and muscle wasting. And that's how we usually see it. How often do we remind ourselves that rapid unintended weight loss is a danger signal and should be investigated as a symptom of cancer or a signal of cancer? So then what actually leads to this weight loss and muscle wasting? Well, we know that there are a number of different things that lead to the weight loss and muscle wasting, the cachexia of cancer. First off, we know that calcium signaling and changes in the mitochondria are actually occurring as a result of the cancer. And you'll see this in the loss of the myofibrils, the fibrils that are actually in our cells. We've talked a lot about an increase in apoptosis and a decrease in regeneration. This is going to cause further wasting because we have a lot of normal cell apoptosis and a decrease in normal cell regeneration. Inflammatory mediators. The inflammatory mediators that we should be thinking about are the pro-inflammatory cytokines. Tumor factors actually have the ability to increase that protein turnover. And by increasing the level of pro-inflammatory cytokines and tumor factors, that increases that protein turnover, therefore muscle wasting and cellular wasting. At the same time, adipose tissue is being broken down. What's also very interesting is our brain pathways are also altered in cachexia. And in those brain pathways, appetite suppression is greater than appetite stimulation. So patients with cancer tend to lose their appetite simply because of these altered brain pathways in the way we respond to appetite. Some other manifestations that we frequently see include fatigue, immune suppression, and some of those systemic effects. So in fatigue, think about how fatiguing chronic inflammation is. Remember, cancer is a pro-inflammatory state. We've talked a lot about nutrition. Both the use of the incredibly high use of nutrients by the cancer cells, as well as those brain pathways that affect the appetite. Those are also going to lead to fatigue. With immune suppression, we think about the effect of aging. Remember that accumulation we talked about earlier? As we get older, we tend to, that accumulation of altered genes gets higher. We also know that as we get older, our immune surveillance isn't as strong. Changes in the T cell pathways that we talked about and the inability or the failure to recognize cancer cells as not good antigens is part of that immune suppression. And then last but not least, when we look at systemic effects, we have to look at the multiple effects of cachexia, inflammation, the inability of the tissues to do their normal work because those tissue cells are completely dedicated to the cancer cell survival. The final characteristic we'll talk about is metastasis. Now, not all cancer cells have the ability to metastasize. We also know that different types of cancer have different patterns of metastasis, so they tend to be somewhat tissue selective. So that the ability to metastasize and where these cancer cells tend to move is something we learn about cancers themselves. So for example, if we look at breast cancer tends to metastasize to the brain and the bone. Colon cancer tends to metastasize to the liver. If you look at overall patterns of metastasis, liver is probably one of the most common sites of metastasis, and this is probably due to the highly vascular and lymphatic supply of the liver. The brain is a less common site of metastasis, and a part of the reason for this may be due to the blood-brain barrier that serves as a stopping point for some cancer cells. Now, in order for a cancer to metastasize, or cancer cells to metastasize or move to distant sites, they have to be able to do some very specific things. First off, they certainly have to be able to outgrow and alter their immediate limiting environment. Now, we know that locally they're very good at that, but can they get even beyond any immediate environment limitation? They have to be able to access local blood and lymphatic vessels. So then they have to be able to detach themselves from the extracellular matrix through secretion of proteases. Certainly they have to be able to survive in the circulation. So once they enter the circulation or the lymphatic system, they have to be able to avoid apoptosis and shear forces. And then last but not least, once they get to that new environment, they have to be able to attach and grow in that new environment. So in some cases they may survive, but they may not proliferate. When they do that, then once again, they're waiting for the environment. They're waiting for optimal conditions. So even though a cell may actually move to a new area, it may be able to support its limited growth in that area. It may not be able to undergo rapid proliferation and cause a second cancer site of metastasis. So what does all of this metastasis stuff and all this cancer stuff mean clinically? Well, the clinical relevance, and especially with metastasis and the growth of cancer cells in general, is to think about staging. The World Health Organization has a staging system, and you've seen this in your practice most likely. But the staging system is the TNM staging system, and this is tumor, nodes, metastasis. And there's a great example of this in your text, and that's figure 12.23, and it kind of walks you through a great example. But when we look at TNM, in general, the higher the number in each staging area, the more extensive the cancer. So let's look at an example of that. So let's say that someone is a T1, N1, M0. So tumor 1, nodes 1, metastasis 0. That's a pretty local, a localized tumor or localized cancer. If you look at a T3, N2, M2, or I'm sorry, M1, you know that it is in more. The tumor is larger, it has invaded lymph nodes, and it has metastasized. How do we also use then this information about cancer cells and about metastasis in our therapy goals? Well, when we look at therapy, we have some very specific goals. We either we may want to remove those cancer cells. That's what surgical therapy does. But not all cells can be surgically removed. Sometimes we can debulk a tumor, or we can remove the tumor in total. Other cancers, like a leukemia, we have no way of surgically removing them. The second way that we provide therapy, or we accomplish therapy, is to stop the cancer cell growth. In order to do that, we may be able to starve them of nutrients, stop the angiogenesis, and some of our cancers chemotherapies are dedicated to exactly that. Certainly, we are happy about killing cancer cells. We can use this through chemical toxins, chemotherapy agents, or through stimulating radiation injuries through either internal or external radiation, because we're facilitating cellular injury through radiation cell injury. Last but not least, we use immune-based therapy. With immune-based therapy, one of our goals is to increase that T cell response. Another goal may be monoclonal antibodies. When we use monoclonal antibody therapy, we are either binding to and damaging the cancer cells. Those antibodies are going to bind to or damage the cancer cells, or may be able to inhibit checkpoints. And when we do that, we allow stronger T cell cytotoxic responses. The final thing that we'll talk about briefly is cancer in age groups. In children are more likely to develop leukemias, which is the most common type of cancer in children, lymphomas, and the second most common is brain cancer. We know that diagnosis of cancer in children is higher during active growth periods, and so we want to be especially vigilant during that time. We talked about, and so when we look at leukemias, we would want to be especially aware of the 6 to 24 month time frame and the adolescence time frame. In elders, we know that the incidence of cancer increases with age. Certainly, cancer in elders is supported by changes in our immune system as we age, in the competence of our immune system as we get older, and that idea of stepwise additive effects, because we've had longer time. As we get older, we had a longer time to be exposed to potential gene mutations. We also know that in elders are more likely to develop solid tissue and organ cancers than they are leukemias and lymphomas. I know we've talked about a lot. I do expect that you'll be able to identify or compare and contrast the differences between cancer cells and normal cells. We know that cancers result from gene mutations that alter the function of cells. We also know that cancers have the ability to change their environment, to change the immune system to its benefit. So we can look at cancer as a failure or the inability of the inflammatory or immune system to not accomplish its self-policing functions. We know that viruses have been implicated in several cancers. We talked very, very briefly about HPV and hepatitis B and C. But understanding how cancer cells behave leads to surveillance, detection, targeted therapies, and recovery. I hope this information has been helpful, and if you have any questions, just let me know. Thanks for your attention.